What are the biomarkers for neuroinflammation in schizophrenia? Have you ever wondered how inflammation in the brain relates to schizophrenia? Understanding the biomarkers for neuroinflammation can provide answers. Neuroinflammation occurs when the immune system responds to injury or infection in the brain. This process can be influenced by genetic factors, environmental triggers, or even lifestyle choices. In schizophrenia, neuroinflammation is increasingly seen as a key player in the disorder's development and progression. So, what exactly are biomarkers? A biomarker is a measurable indicator that reflects a biological condition or process. In the case of schizophrenia, biomarkers for neuroinflammation help signal ongoing inflammation in the brain. Let's look at some key biomarkers that researchers have identified. First up are cytokines. These small proteins are released by immune cells and play a role in regulating inflammation. In individuals with schizophrenia, elevated levels of specific cytokines like interleukin-6, tumor necrosis factor alpha, and interleukin-1 beta have been found in both blood and cerebrospinal fluid. This is especially true during early episodes of psychosis and in chronic cases. Next, we have C-reactive protein. This substance is produced by the liver in response to inflammation. High levels of C-reactive protein in the blood are linked to increased neuroinflammation and poorer clinical outcomes in schizophrenia. Another important marker is related to microglial activation. Microglia are the immune cells of the brain. When they become activated, they release inflammatory substances. Researchers can detect this activation through advanced imaging techniques or by measuring specific proteins in cerebrospinal fluid. Gene expression patterns also play a role. Certain genes associated with immune function and inflammation show different levels of expression in individuals with schizophrenia. This is particularly noticeable in brain regions that are important for language, memory, and sensory processing. Lastly, neuroinflammation can lead to structural changes in the brain. For instance, reduced gray matter volume, especially in the hippocampus and cortex, can be observed through neuroimaging techniques like magnetic resonance imaging. Understanding these biomarkers is not just academic. They have practical applications in the real world. Clinicians can use these indicators to identify individuals at risk for schizophrenia, monitor how the disease progresses, and evaluate treatment effectiveness. For example, measuring cytokine levels or C-reactive protein in blood samples can provide early warning signs of neuroinflammation. In summary, biomarkers for neuroinflammation in schizophrenia include elevated cytokines, increased C-reactive protein, microglial activation, altered gene expression, and structural brain changes. These markers can be measured in blood, cerebrospinal fluid, or through neuroimaging. Recognizing these biomarkers can aid in early detection, monitoring, and targeted treatment strategies for schizophrenia, especially for those at risk.